Hi, everyone. So now I will start a new topic and I will introduce about metaproteomics. So first of all, what is metaproteomics? If you look at the first part of this word, meta originates from Greek, and unfortunately, I don't know how to pronounce it properly. So it meaning after or beyond in Greek. And if you check the meaning of it from Merriam-Webster, it says you usually use together with the name of a discipline to describe a new discipline, which is related to the previous one, but is more comprehensive. So what is metaproteomics? It is the study of all proteins in microbial communities in microbiota. Sorry, there's some typo. So what is microbiota? Now, microbiota is a very hot topic, and it refers to the diverse community of microorganisms. And you can see it everywhere, like in environment, in our bodies, and also in some man-made environment. And microbiota play a very crucial role in maintaining the host health, like the gut microbiota help us keep the homeostasis of our health, and it's not only related to the gut health, but also related to brain, to your heart, almost everywhere, and also in the environment, they play an essential role to keep the ecosystem balance and promote the biodiversity. And before I start to introduce metaproteomics, I would like to talk about metagenomics first, because actually metaproteomics learned a lot from the metagenomics. And before the development of metagenomics, people study the microorganisms in a very traditional way. So basically what people do is to culture all the microorganisms in the lab and isolate like each species or each strain and sequence them. And at the very beginning, they just sequence 16S RNA part, which is just a small part in their genome. And first of all, we can take a look at the first part of the experiment is to culture the microorganisms and isolate each of them. First of all, it's quite low throughput, so it takes a lot of time to find the best conditions to let as many microorganisms as possible to stay alive, and you have to isolate them, you have to extract the DNA, so it's very low throughput. And also bias is actually introduced during the bacterial isolation, because you can barely find the proper condition that all the microorganisms from the original environment can survive there. And also because for microbiota, this microorganism actually have a lot of interactions. So some microorganism can not really live without the other ones. So in this case, you lose a lot of information, and because you select certain ways to culture your microorganism, you introduce some bias to your samples. And also the coverage is very low, because actually for most of the organisms in the nature, you cannot really find the proper way to culture them in the lab. So you don't have enough information. And for the second part of the experiment is to extract the 16S RNA and determine the composition of the microbiota. It's also not good enough, because first of all, 16S RNA cannot reliably classify bacteria at lower taxonomic levels. What means that is you can use this molecule to distinguish microorganisms that are really different from each other. Like at the genus level, you can see clearly difference. But for some microorganisms, for example, like different E. coli strains within the E. coli species, for some of them, you cannot really distinguish them within this 16S RNA sequence, which is quite small if you're comparing it with a whole genome. And also for 16S RNA, it only provides you the information about the composition of your samples, but you don't really know what can they do. So in terms of function, so in 1998, it's very clever people, they create the term metaproteomics, and it was used to refer to the idea that a collection of genes sequenced from the environment could be analyzed in a way that like the study of a single genome. So for metagenomics, you don't really like need to isolate them. They thought we can, okay, just remove some like, in this case, soils, some big particles that don't belong to the microorganism. They just separate all the bacteria, and I don't care who they are. I just extract all the DNA from them, and I sequence all of them, and I treat them as a whole genome and do some downstream analysis afterwards to distinguish who they are and what can they do. So actually, back in that time, it was really like an ambitious picture of how it will be in the future, because in 1998, people are still struggling in DNA sequencing. And for the Human Genome Project, it started in 1990, and people around the world spent billions of dollars just to get the whole genome, human genome sequencing. And the draft of Human Genome was published in 2001. And so they really like ambitious to dream in 1998, and it actually becomes true in less than 10 years. So here you can see the workflow of metagenomics in modern ages, and if you compare it with the previous one, there is no big difference. So basically, what we usually do is to isolate the microbes, and we extract the DNAs and do the DNA sequencing, and at the end, we analyze the data to determine who they are and what can they do. So for metaproteomics, because metagenomics, for researchers in metagenomics, they already spend a lot of time and money to optimize all the part of the analysis. So for metaproteomics, we already got a lot of experience. So people start to use the term metaproteomics in 2008 to describe the proteome from the mixed microbial communities. And they published one paper, and if you compare the workflow between the metaproteomics and metagenomics, they look almost like the same. It's just the material comes from DNA to protein, and for metaproteomics, we extract all the proteins and we measure them in mass spectrometer, and at the end, we do the analysis to get the information who they are and what are they doing. So what's the advantage of metaproteomics? So basically, it's also the advantage of proteomics over genomics, because they present active function instead of functional potential. For at the DNA level, you can detect DNA, but you don't really know if this gene are actually active and translated into proteins at the end. And also they can show faster changes in response to stimuli. So especially for microorganisms, sometimes you don't really see abrupt changes at the DNA level. And also for metaproteomics, a very good thing is you can analyze microbial proteins and host proteins altogether. Now I'm talking about like gut microbiota. So in this case, it will really help you to also understand how the microorganisms interact with the host as well. But it also has some disadvantages. First of all, the detection sensitivity is lower than the metagenomics. That's also because of the difference between the genomics and proteomics. So for genomics, we have PCR, so we can kind of amplify the signals even they're super tiny. But for proteins, at the moment, it's not possible. So the detection sensitivity is relatively lower. And also the classification can not really reach lower taxonomic levels. That's also because of the difference between the protein and the DNA. So this is called codon degeneracy. So basically, when you translate the RNA into the peptide, you use three nucleotides will decide which amino acid will they create. And from this figure, you can actually see that for most of this, for all of this amino acids, you can create them with different combinations of nucleotides. So in this case, you can see that actually this is because of the redundancy of the genetic code, which is also called synonymous mutations. So for some species, for some different species for some different strains under the same species level, for some of them they could have some difference at the DNA level. They can have certain nucleotide changes, but when they are translated into the peptides and becomes protein, they are actually the same. So in this case, you cannot really distinguish certain proteins if they are from this strain or from that strain. But we also have another way to solve this problem. So there is an algorithm called lowest common ancestor. Basically, this conception is very similar to the conception of protein groups. So yesterday, Pelly already introduced how we put two different proteins that share same peptide into the one protein group. So in this case, we are following the same way to do it. If one protein or one peptide is shared by two different strains, let's say E. coli K12 and another E. coli strain, so we don't care where exactly does it comes from. We can just go up to the species level. If it's exclusively in E. coli, then we can just tell, okay, this peptide or this protein is from E. coli, so we don't really care which specific strain it is. So before I compare the metagenomics and the metaproteomics, and they have the meta part in common. And next, I would like to introduce the difference between the metaproteomics and traditional proteomics, which is focusing on, most of the case, focusing on one single or very few organisms at the same time. So the first difference is at the sample preparation part. So this is the slide I took from my first presentation. So in proteomics, you extract the proteins from your samples, but it do make a difference between the samples, because if we are studying some cells or organs from the animals, actually the structure of the cell membrane is different. So on the right, you can see this is a classical structure of the animal cell membrane. So you just have two lipid bilayers, so which is quite easy to break. So in the experiment, you can just use ultrasound to break it. But for bacteria, the structure of the cell membrane is different. They have an extra sugar part, which is kind of like cell wall in the plant. So in this case, if you want to extract all the proteins from the microorganism, you need to use more extreme ways to break all the membrane and cell walls of all different kinds of microorganisms. And later, next I would like to focus on a very big difference between the metaproteomics and traditional proteomics. So this is the figure from Walter's introduction about the Andromeda score yesterday. So basically for proteomics study, you give the software, you upload the software, your raw data that was measured from the mass spectrometer, and to identify the proteins, and peptides, you also need a database for the database search. So for single organism, if you are working on human or mouse, it's super easy. You just go to UniProt, like Dima showed yesterday, how to download the database and use that to do the database search. But the problem for the microbiota is you don't really know the composition of your sample, so you don't know who they are. So in this case, for the database search, if you don't put the sequence that's actually exists in your samples, you have no chance to identify them. But if you put all the sequence of them, all the sequence from the UniProt, it's huge, and for sure you will put a lot of redundant information that will not help you identify your proteins. So there are several different strategies how to select the database. The first of all is use a public database without selection. For example, you put the keywords bacteria and download all the bacterial proteins from the UniProt, which is not really practical. So for example, I show you this number that I checked yesterday for the E. coli K12. In UniProt, you can find 6,000 proteins, but if you use the keyword UniProt bacteria, you get 153 million proteins, which means you will have an extremely long running time. And at the same time, you will have super high false positive rate, which will finally lead to a very short list of identification. And I will quickly show you an example how it looks like. So this is the proteomics data from the mouse, the rat brain, from 3 different rats, and they were measured. And if we want to do the database search, of course, we will just use UniProt rat sequence to do it. But in this case, I would like to show you like the negative part of putting too many redundant sequence in the database. So I did another database search. So in this case, I put a lot of sequence from the bacteria as well, which is almost 1 million sequence. So when we do the database search against this database, the size of the database is almost like 100 times greater. So for the original one, you have the target database, which is the size of the rat, and when you reverse it, and you have the decoy database as the same size. And that's the same case if you do the database search against the rat with a bacteria database. So the size is like 100 times greater. So after you do the database search, actually you can really see like how much are the positive and negative identifications, because in the rat brain, you actually don't expect to see bacterial proteins. So in this case, if you see the identification of reverse rat proteins, of course, it's incorrect because it's a reverse data. And if you see bacterial proteins, then it's also incorrect because it's from the rat brain. And of course, if you get identification in the reverse bacteria, it's also incorrect because it's the reverse database. So how the data will look like. So first of all, we don't do any FDR control. We don't care about the decoy database. Then we do the database search. We get around the same level of PSM identification, but we have to do the FDR control. And if you use by default 1% FDR, you could see that if you search against a database with rat and also bacteria, you get only around half of the identification. And what's going on there? If you plot the histogram of all the identified PSM, you can see that on the left side is the results that when you do database search only against the rat database. So when you do FDR control, you rank all the identifications, and you just count the ratio of the decoy matches, and once it reaches 1%, it will stop. And in this case, the red line shows the cutoff of FDR1 stopped at almost 305,000 rank. But when we search against a huge database that's including a lot of redundant, you can see that the cutoff of the FDR 0.1 stopped at around 2,600. And as I showed you, that's in the second test, when we search against a huge database that also include bacteria, we can actually spread the result because we know some of them are from bacteria. And from this one, when we do the FDR control, we just care about the target decoy. And when you look at this figure, and you can see that the most contribution of the false negative is actually not from the reverse part of the red, but it's from the reverse part of the bacteria. So this showed you what is a negative effect of the redundant proteins in the database. If you really put a lot of proteins that in theory should not present in your database search, it's gonna take a very, very long time. And because you have a lot of redundant, all this redundant part will not help you to identify more proteins because they are not there. But when you revert all the sequence, you get a huge decoy database, then the probability to get a high scoring false decoy match is very high. So in this case, if we take a look at the detail of how this PSM comes from, you can see for the reverse part, almost all of them comes from the bacterial part, the redundant part. So we don't really recommend you to use all the proteins from the public database without any selection. Then there is another method to overcome this problem, is to select certain proteins from certain microorganisms. So first of all, they will identify the microbial composition based on high abundant proteins, which is in short HAPs. So what is HAPs? So HAPs are basically the most abundant proteins, which are mainly ribosomal proteins and elongation factors, because they are the key proteins that plays a very important role in the protein translation. So for all of this active microorganisms, these are the most abundant proteins, and they account for around 20% of cell proteins by weight. So it gives us a very strong signal to detect the composition of them. And this is a workflow, how it works. So basically, some people already create a database that include all the HAPs, high abundant proteins, into one database. So first you do a database search against this database and this. So the purpose of this first database search is knock knock, who's there? And with this information, get a list of microorganisms. Then you extract the proteins from certain organisms from the big database. Then you do the second round of search, which is very good. And which is even better is in MaxQuant, there is a very special feature in the identification called split protein groups by taxonomy ID. So this was actually not initially developed for metaproteomics, but for some benchmarkings, for example, you will mix several different species to do some benchmarking study. So as Pelly introduced yesterday, we will merge the proteins that share same peptide into protein groups. So in this case, if you know that a mouse protein and the E. coli protein share the same peptide, it doesn't really make sense to put them into the same protein groups to do the quantification, because they actually should not be one protein groups because they are from different organisms. So in this way, if we select this option, and when you put all the the proteomes from different microorganisms, when we merge them into protein groups, they will, the proteins from different organisms will not be merged together. And this is very good. It's sample specific and the size is acceptable. But it's also a problem for some researchers who is focusing on environmental samples, because for environmental samples, they are not really very well studied for these organisms, so you don't really get a lot of information from it. But for these people who are working on human gut microbiota, mouse microbiota, there were a lot of genomic study on it, so you get a lot of information about each microorganism already. So this, if we go back to the comparison of the metaproteomics and metagenomics, so you can see there are some disadvantages of metaproteomics. So if we see it in another way around, actually these are the advantages of metagenomics, and we can make use of this advantage to create a very sample-specific database. So in this way, we just use the same sample, we do the genome sequencing, and we use a metagenome to translate it into the protein sequence, and we can use this metagenome database as a sample-specific database, and also the size is really under control. And even better thing is it's suitable for all microbial data. So even if you are working with environmental samples that don't really have a very nice reference database, it's also fine. So there are also some interesting topics in metaproteomics. I will just mention it, but I will not spend time, like the DIA analysis with metaproteomics data. So for MaxQuant, we already tried to use on-fly prediction mode to run DIA data with metagenome database. It works very well, and it's at the same level as other software that were focusing on the DIA analysis. And also because we don't really know the composition of the samples, so it's challenging to create a database. So some people are trying to figure it out in another way. They use de novo sequencing, which is another strategy that calculates the sequence of your samples, not based on the database search, but based on the spectra itself. And also, some people are developing some deep learning model to improve the identification in metaproteomics. Some of them were designed just for metaproteomics, some of them are not. And I believe tomorrow, this Tesserae lecture might introduce something about metaproteomics, because I see that in your manuscript, you show a result how you can improve the database searching in metaproteomics. Okay, that's all. Thank you very much.